Good afternoon. Welcome to this session of National Workshop on CSIR-UGC-NET-JRF Lectureship Preparation in Life Science. Myself, Dr. DEVI CHINMAYEE, Assistant Professor, working in Sree Ayyappa College for Women, Nagarkovil, Tamil Nadu. Today, I am going to present before you the topic called bioremediation. Bioremediation. It is one of the green technology used for cleaning our pollutants. It refers to use of either naturally occurring or deliberately introduced microorganisms to consume and break down environmental pollutants in order to clean a polluted site. It is a process that uses mainly microorganisms but also plants or microbial or plant enzymes to detoxify contaminants in soil and other environment. It may be the contamination in water, air, etc. Bioremediation has been identified as a biological response to environmental abuse. Due to anthropogenic activities, that is human activity, our environment is loaded with tons of pollution that can be removed by using living organisms such as microbes, plants, etc. This process is called bioremediation. It refers to the process of using microorganisms to remove the environmental pollutants or prevent pollution. The removal of organic waste by microbes for environmental cleanup is the essence of bioremediation. The other name used for bioremediation are biotreatment, bioreclamation, and biorestoration. The pollutant generally seen in the environment is called xenobiotic. It is broadly referred to the unnatural foreign and synthetic chemicals such as pesticide, herbicide, and other organic compounds which are released to the environment due to anthropogenic activity. So, coming to principle of bioremediation, and it depends on stimulating the growth of certain microbes that use contaminant as their like oil, solvent, and pesticide as a source of food and energy. So these microbes use this contaminant as their food and use it for energy. These microbes consume the contaminant and convert them into small amount of water and harmless gases like carbon dioxide. So they use the pollutant which are seen in the environment and convert them into harmless substances into environment. Effective bioremediation need a combination of the right temperature, nutrient, and food. Otherwise, it may take much longer for the cleanup of contamination. So, the main principle of bioremediation is converting contamination into harmless substances in minimum period of time. If conditions are not favorable for bioremediation, they can be improved by adding amendments. So these are the substances such as molasses, vegetable oil, or simply air which increase the growth of microorganism. So if the microorganism is not properly cleaning the pollutant, we can add certain substances that increase the growth of microorganism, and during their growth they can also convert the contaminant into harmless substances. So these amendments create optimum condition for microbe to flourish and complete the bioremediation process. The process of bioremediation can take anywhere from a few months to several years. So main objective of modern bioremediation technique is to reduce this time and convert a large amount of contaminant into harmful substances. The amount of time required depends on variables such as the size of the contaminated area, the concentration of the contaminant, conditions such as temperature, soil density, and whether bioremediation will take place in situ or ex situ. So there are several factors which depends the pace and rate of bioremediation. The factors are the contaminated area, the length or size of contaminated area, the concentration of contaminant, temperature, if it is happening in soil, bioremediation which is happening in soil, then soil density and the soil pH, all the matter which will affect the bioremediation. So bioremediation is a triple corner process, that is, the pollutant, the environment and the organism. These three depend upon the bioremediation. So these three are the major factors which are seen, regulating bioremediation process. So coming to pollutant, pollutants may be inorganic or organic. It may be solid, liquid or gas. So the bioremediation process can take place in soil, water or air. So the major environment where the bioremediation taking place are soil, water and air. So major organism used for bioremediation are microorganism, bacteria, fungus, etc. and also several species of plant which are used for remediating the pollution from the environment, and also several enzymes which is secreted by these microorganism or plants can be also used for converting the harmful pollutant to harmless one. So coming to categories of bioremediation. Biological remediation can be categorized into two types, that is microbial remediation and phytoremediation. In the present session, we will look forward in microbial remediation. Phytoremediation we will see on next section. Microbes may be bacteria or fungus. The microbes for the which is used in the bioremediation process may be bacteria or fungus. Although many microorganisms are capable of degrading crude oil present in soil. Microorganisms are well known for their ability to break down a huge range of organic compounds which is present in our environment as a pollutant and absorb inorganic substances. Currently, microbes are used to clean up pollution treatment in process known as bioremediation. Different microbial system like bacteria, fungus, in fungus also yeast, actinomycetes can be used for removal of toxic substances. So these several organisms, several microbes can be used for action against pollutant present in the environment. Microorganisms are readily available, rapidly characterized, which show high diverse and which is omnipresent, present everywhere and can use many noxious elements as their nutrient source. So we can't grow, the microorganism can grow in everything, and even may in many pollutants these microbes can utilize that chemical as their source of food and convert them into harmless substances. They can be applied in both in situ and ex situ condition. In addition, many extreme environmental conditions can be cleaned by such microbes. Such microbes can clean several extreme pollutants which is present in the environment. Although many microorganisms are capable of degrading crude oil present in soil, it has been found beneficial to use a mixed culture approach than a pure culture for bioremediation. So we can mix several species of microbes and culture it in that toxic pollutant. Different bacteria can be used for removal of petroleum hydrocarbon contaminant from soil. So several bacteria can be used. So they include Pseudomonas, Aeromonas, Moraxilla, Nocardia, etc. So these many species of bacteria can be used against major pollutants seen in environment.
Next, coming to type of bioremediation. There are many types of bioremediation. The major ones are biostimulation, bioaugmentation and intrinsic bioremediation. Biostimulation. In biostimulation, it is a method in which bacteria are motivated to start the process of bioremediation. So first, the bacteria will get a motivation in the form of important nutrients, and then it will start bioremediation. So at first, release of nutrients and other important substance in the soil where there is need or removal of contamination is required. First in the contaminated site, we will release some nutrients which are important for the growth of the bacteria. Then the bacteria is cultured. This nutrient which is released is in the form of gas or liquid, and it increases the growth of bacteria in that area. So in the contaminated area, due to the high nutrition, the bacteria will grow. As a result, bacteria and other microorganisms remove contaminants quickly and effectively from that soil. So this process is called biostimulation. So this process is called biostimulation. Next, coming to bioaugmentation. Microorganism that can clean up a particular contaminant is identified. So a microorganism which will clean a particular contaminant is identified and added to that contaminated site. So if a soil, for example, if a soil is contaminated by a particular type of heavy metal, lead, so we identify a bacteria which will remove the lead from that soil. So then releasing that bacteria into lead contaminated soil is called bioaugmentation. Microorganisms that clean up a particular contaminant are added into the contaminated soil and water. Bioaugmentation is more commonly and successfully used as contaminant removed from the original site, such as municipal wastewater treatment facility, etc. So the bioaugmentation technique is a particular type of bacteria which will act on particular type of contaminant. So we can release that bacteria into that contaminant and remove that contaminant from the environment. Is called bioaugmentation. Next, intrinsic bioremediation. It is a process which takes place in soil and water because these two places are always full of contaminant. This process is also called as natural attenuation. So it means use of microorganisms to remove the harmful substances from soil and water, especially those sites which are treated with this method are underground. For example, underground petroleum tank rupture and such places, this intrinsic bioremediation is used. So generally used in a site where toxins are present, mainly in soil and water, maybe in underground soil contaminant or underwater contaminant can be removed through intrinsic bioremediation. So it is a natural attenuation. So use of microorganism to remove harmful substances from soil and water which cannot be removed by human being in normal cases. So that can be removed by the bacteria. So coming to bioremediation strategies. So there are generally two strategies for bioremediation, that is in situ bioremediation and ex situ bioremediation. So in situ bioremediation means bioremediating a particular contaminant which is present at a particular site. So we are releasing bacteria or any other living things to this particular site where there is contaminant is present. Ex situ bioremediation is taking the contaminated soil or water into a particular place where bioremediation is taking place. So in situ bioremediation is the bioremediation at the particular site, and ex situ bioremediation is the bioremediation which is away from the site, actually the some special place where bioremediation is taking place. The basis of removal and transportation of waste for treatment basically are of two methods. They are in situ bioremediation and ex situ bioremediation. In situ bioremediation, it involves direct approach for the microbial degradation of xenobiotics at a site of pollution. Where there is polluted, if a particular area is polluted by oil spill, the soil is polluted, we will release the bacteria or a microorganism into that soil where there is polluted. Such type of bioremediation is called in situ bioremediation. So generally there are two types of in situ bioremediation, that is intrinsic bioremediation and engineered bioremediation. So it has been, this in situ bioremediation is successfully applied for cleaning up of oil spillage, beaches, etc. for cleaning the pollutant which is present in that particular environment. So in situ bioremediation is when the contaminated site is cleaned up exactly where it is occurred. So where some place where their pollution occur is cleaned by the microorganism in that polluted site itself. There is no need to remove soil or transport soil or water in order to accomplish remediation. For our remediation, we should not. In situ remediation, there is no need to take the soil, water or any other polluted thing into a particular place where is bioremediation taking place. So it is the bioremediation of soil or water at the place where it is polluted. In situ biodegradation involves supplying oxygen, nutrients by circulating aqueous solution through contaminated soil to stimulate naturally occurring bacteria or any other genetically modified bacteria to degrade organic contaminant. It can be used for soil and groundwater. Soil and groundwater contamination can be removed by in situ bioremediation if the condition there, if the in the polluted site, if the condition is not favorable for bacteria, we can supply some nutrients or oxygen to that contaminated site to increase the growth of bacteria and providing bioremediation in that area. It is the most commonly used type of bioremediation because it is cheapest and more effective. So it is generally better to use in situ bioremediation. So there are generally two types of in situ bioremediation. They are engineered bioremediation, that is intentional changes. Engineered bioremediation, there is intentional changes, and intrinsic bioremediation simply allow biodegradation to occur under natural condition. So intrinsic bioremediation, we have to do nothing. Naturally it will bioremediate. That polluted site will be cleaned within a span of time. So intrinsic bioremediation is doing nothing, just leaving the place, and the bioremediation will take place. So engineered bioremediation is intentional change. We can do several changes in that area and introducing several new microbes into a particular area which is not actually present there. We can introduce certain microbes and bioremediate. So first coming to intrinsic bioremediation. It is a bioremediation under natural condition. So use of microorganism already present in the environment to biodegrade harmful contaminant. So if a site is contaminated, if we leave that site as such, it will be remediated, but it will take more time than if we are using a particular type of microbes. If the microbe which are capable for remediating a particular contaminant, if we identify which contaminant is present in that area and introducing that particular type of microbe which will degrade that contaminant, then we can increase the pace of bioremediation. So intrinsic bioremediation is a natural bioremediation in which the use of microorganism which is already present in that environment to biodegrade harmful contaminant. There is no human intervention involved in this type of bioremediation. So it is a natural process. Since it is the cheapest mean of bioremediation available and most commonly used. When intrinsic bioremediation is infeasible, scientists turn next to engineered bioremediation. So if a contaminated site is kept as such for the remediation, if it is not possible, the scientists will find some other way to remediate a particular soil or water. So that is engineered bioremediation. So second approach involves the introduction of certain microorganism in the site of contamination. When site conditions are not suitable, engineered system have to be introduced to the particular site. So if it is not suitable for a normal natural remediation, we can introduce several other type of microbes which are specialized for that particular contaminant. Engineered in situ bioremediation accelerate the degradation process. It will increase the pace of bioremediation by enhancing the physicochemical condition to encourage the growth of microorganisms. Oxygen, electron acceptor and nutrients, that is nitrogen and phosphorus, promote microbial growth. So during engineered bioremediation, particular type of microorganism which is specialized for that contamination is introduced into the site where there is contamination, and if there is if there is a nutrient supply is very less at that area, we can supply nutrients, oxygen, nitrogen, phosphorus, etc. for increasing the microbial growth. So, in situ engineered bioremediation type involve bioventing, another process. So, there are different types of in situ engineered bioremediation. So, first one is bioventing. It involves supplying air and nutrients through wells to contaminated soil to stimulate the indigenous bacteria. So, the contaminated soil is supplied with nutrients and air which is required for the growth of bacteria, that is indigenous bacteria, and stimulate its growth and stimulating bioremediation process. This process is called bioventing, that is wells are created. Through wells we will supply contaminated soil. Into the contaminated soil we will supply air and nutrients which are necessary for the growth of bioremediating bacteria. Then this bioremediating bacteria convert this contamination into non-toxic compounds. So next one, biosparging. It involves the injection of air under pressure below the water table to increase groundwater oxygen concentration and enhance the rate of biological degradation of contaminants by naturally occurring bacteria. So, biosparging, another technique which involves injection of air under pressure below the water table. So, into the well we will a large well, we will inject some air under pressure below the water table to increase groundwater oxygen concentration and enhance the rate of biological degradation of contamination which reach the water table. This biosparging is done using the naturally occurring bacteria. So, bioaugmentation, we already said the bioaugmentation, it involves practice of adding specialized microbes on their or their enzymes for the in the contaminated site for removing the contamination. So it involves practice of adding specialized microbe or their enzyme preparation to polluted site to accumulate transformation or stabilization of specific pollutant. So a bacteria which is specifically affecting a particular type of contaminant is added in the bioaugmentation process. So next, coming to ex situ bioremediation. So ex situ bioremediation is taking the waste toxic material including soil or water into another site which is not originally polluted site. We will take the water or soil or contamination to another site and then bioremediating. This is the process of ex situ bioremediation. The waste or toxic material can be collected from the polluted site and the bioremediation with the request microorganism can be carried out at a designed place. For example, in a bioreactor or particular place. Metabolic effect of microorganism on xenobiotics. The major metabolic effects are detoxification of xenobiotics, degradation, breaking down of xenobiotic, activation and conjugation. These are the four effects microorganisms cause on the pollutants or xenobiotic. Type of reaction in bioremediation. There are three types of reaction occur during bioremediation. They are aerobic, presence of oxygen, anaerobic, without oxygen, and sequential. Aerobic and anaerobic process occurs sequentially, that is sequential bioremediation. So ex situ bioremediation are of three types: aerobic, anaerobic, and sequential bioremediation. Okay, so next coming to ex situ engineered bioremediation strategies. So when a leaking or contamination of sites occur, the contaminated soil or water can be taken to some other site, and then bioremediation taking place is the ex situ engineered bioremediation. So the contaminated soil is excavated and treated in number of ways. First way is the slurry method, in which the contamination is mixed with water, oxygen, and fertilizer to stimulate microbial degradation of pollutants. The second method is solid phase bioremediation. In solid phase bioremediation, we can compost the soil with the hay or other bulky agent and mix together. The second method is land farming, and third is biopile. Let's go into detail of these processes. So solid phase system in ex situ bioremediation, first method is composting. All of you know the composting, which is the technique that involves combining contaminated soil with organic compounds such as agricultural waste. So we will first mix the contaminated soil with agricultural waste, which will act as the nutrients for the bacteria which is doing bioremediation. The presence of these organic materials support the development of a rich microbial population and elevated temperature characteristics of composting. So after composting, this microorganism will convert the polluted contaminant into harmless substances. So next coming land farming operation. Land farming is a simple technique in which contaminated soil is excavated and spread over a prepared bed and periodically tilled until pollutants are degraded. So first the soil which is contaminated is taken from the original polluted site, and then it is spread in a prepared bed and periodically tilled until pollutants are degraded. This practice is limited to the treatment of superficial 10 to 35 centimeter of soil. So it is only applicable for the top soil. Okay, so the third method is biopile system. Biopiles are a hybrid of land farming and composting. Essentially, engineered cells are constructed as aerated compost piles, typically used for treatment of surface contamination with petroleum hydrocarbon. They are a refined version of land farming that tend to control physical loss of contaminant by leaching and volatilization. Biopile provide a favorable environment for indigenous aerobic and anaerobic bacteria. So this is the hybrid technology biopile system of land farming and composting. So the contaminated soil is constructed in an aerated compost pile. Compost piles are created using contaminated soil and also using various plant waste and animal waste are used for composting, typically used for treatment of surface contamination with petroleum hydrocarbons. Biopile provide a favorable environment for indigenous aerobic and anaerobic microorganisms, which will bioremediate the contaminated soil or material which is present. So next, bioremediation using bioreactor. So contaminated soil is taken and it is mixed with water and oxygen and making a slurry. Then it is put in a bioreactor which consists of several bacteria which bioremediate the contaminants in the contaminated soil. Okay, so this is the method of bioremediation using bioreactor system.
Okay, so if the solid waste are used, it is mixed into water and make a slurry. Then clean soil can be recovered from the bioreactor after bioremediation. So biodegradation of hydrocarbon and petroleum. So the friendly bacteria in spillway product eat oils and hydrocarbon waste. This microbe digest and metabolize these waste, turning it into water and harmless gases. Finally, the microbe release the water and gas back into nature. Hence, the hydrocarbon can be removed from the natural. Coming to advantages and disadvantages of bioremediation. Bioremediation is a natural process and is therefore perceived by the public. Bioremediation is useful for a complete destruction of a wide variety of contaminants. Instead of transferring contaminant from one environmental medium to another, for example from land to water or air, the complete destruction of target pollutant is possible by bioremediation. Bioremediation can often be carried out on site often without causing a major disruption of normal activity. So since it is a natural process, we can carry it out bioremediation without any disruption of normal activities. Bioremediation can prove less expensive since in many bioremediation techniques we are using natural bacteria which are present in the environmental condition. So it is less expensive than other technology that are used for cleaning up of hazardous waste. So low cost, minimum site disruption. Due to bioremediation, other side effect is not caused into the environment. So it is having a minimal site disruption. Simultaneous treatment of contaminated water and soil. So we can simultaneously treat water and soil during bioremediation. Minimal exposure of public and site personnel. So it requires very minimal exposure of public and site personnel. Useful for the complete destruction of a wide variety of contaminant. We can completely destruct or reduce the contamination, wide variety of contamination from every group of chemicals. Can often be carried out on site, often without causing major disruption in normal activity. So can prove less expensive than other technology. So coming to disadvantages, hence many advantages are seen for bioremediation. There are many disadvantages also seen for bioremediation. The first one, the bioremediation is limited to those compounds that are biodegradable. So normal bioremediation is for biodegradable compounds. Not all compounds are susceptible to rapid and complete degradation. Every chemicals cannot be degraded with the microbes or any other living organism. There are some concern that the product of bioremediation may be more persistent or toxin than parent compound. When the microbe are converting a compound into another form, it may be more persistent or toxic than parent compound. So it should be kept in mind before doing bioremediation. So we have to check what the product the bacteria is producing before we are implementing it in the field. Biological processes are often highly specific. Microbial population, suitable environmental growth condition and appropriate level of nutrients and contaminants are required. That is highly specific biological process. So several microbes require a specific environmental condition or growth condition for its proper growth. It is difficult to extrapolate from bench and pilot scale studies to full scale field operations. So when we are doing it in a bench study or if we are doing in a small amount study or small scale studies, so it cannot be applicable to full large scale studies. Okay, so bioremediation often take longer than other treatment options. So the time is the most disadvantage of bioremediation. Take more time. Since it is an eco-friendly process, it requires more time to degrade the contaminant from complex to a simple non-toxic form. So next process which is correlated to bioremediation is phytoremediation. Majorly a branch of bioremediation is phytoremediation. That is remediating the soil, or using the contamination of soil can be remediated using plant. So it can be discussed in next session. So thank you for your patience. Thank you.